What is an epitope on an antigen? Imagine the immune system as a highly skilled detective, always on the lookout for clues that reveal who is a threat and who is not. One of the most important clues it uses are tiny molecular markers called epitopes. These are specific, small regions on the surface of an antigen that the immune system recognizes and binds to. Think of an epitope as a unique fingerprint or a tiny flag that signals, "Hey, I am foreign and need attention." An antigen itself can be a large, complex molecule like a virus protein or a bacterial component. But the epitope is just a small part of that larger molecule, usually about 5 to 15 amino acids long or a few sugar residues. These tiny regions are the exact spots where antibodies or immune cell receptors, such as those on B cells and T cells, latch onto. When they do, it triggers the immune response, alerting the body to attack or neutralize the invader. From the immune system's point of view, epitopes are like molecular ID badges. They help the immune cells distinguish between the body's own molecules and foreign substances like viruses or bacteria. This recognition is very specific, much like a lock and key. The immune receptors are designed to fit perfectly with the epitope, ensuring that the immune response targets only the harmful invader and not the body's own tissues. There are two main types of epitopes. The first is called a linear epitope, which is a continuous chain of amino acids in the antigen. These are recognized by immune receptors regardless of how the antigen folds. The second type is called a conformational epitope. These are made up of amino acids that are not next to each other in the sequence but are brought together in space when the antigen folds into its three-dimensional shape. Recognizing conformational epitopes depends on the antigen maintaining its natural shape, which is important for immune responses and diagnostic tests. Understanding epitopes is key to many aspects of immunology. They guide vaccine development by showing which parts of a pathogen to target. They also help create diagnostic tests that detect specific immune responses and develop therapies like monoclonal antibodies that target disease molecules with precision. In simple terms, an epitope is the exact molecular site on an antigen that the immune system spots and binds to. This tiny region plays a big role in how our immune defenses recognize and respond to threats, keeping us safe from infections and diseases.